Get ready to enter a brave new world with your host, Vasant Dar.
Brave New World is supported by the Center for Data Science, or CDS, at NYU. If your organization is interested in engaging with CDS through student projects, please email cdsindustry@nyu.edu. For more color on the podcast and additional commentary, please subscribe to my newsletter at vasantdar.substack.com. Hello, and welcome to Brave New World. My guest today is computational biologist Rich Bonneau, who is a professor of computer science and biology at NYU. Rich inhabits the intersection of biology and machine learning, which is on fire these days. Rich is also co-founder of Prescient Design, which was acquired last year by Genentech, so he straddles the worlds of industry and academia. That also means he's got to make his research actually work, which is always exciting. Rich, welcome to Brave New World. I am delighted to have you on the show.
Well, thanks for having me, Vasant.
So, Rich, let's start with your background. You're one of the rare people who has this dual background in biology and computer science, which is relatively rare, just given the way academia is and people grew up in silos. How did it happen? How did you sort of end up with this kind of background?
Well, I wanted to be a botanist, and it turns out I'm really bad at dissecting grape embryos out of these woody little grape seeds. So my first projects in the lab, the people running those labs pretty naturally pushed me towards the more technical things. It was pretty natural. So I started off working in botany, got more into molecular biology. Then I joined an NMR lab, and pretty soon we were designing instruments and writing algorithms to pull data and analyze data off those instruments. And so for me, it was actually a pretty natural progression. I've always been interested in computer science and math, but the application and the part of the natural science world that's interested me most has always been biology. So I tried to be a biologist and got pulled more and more into the high-tech part of biology.
So how did you, where was this? Where did you get started interested in botany?
So I actually started working with a number of people at Florida State, and they were interested in plant histones. And that didn't last super long. The lab next door was a guy named Tim Logan, who was an early protein folder who studied how proteins fold with nuclear magnetic resonance. And so there you can watch hydrogens exchanging between the water and the protein. You can watch as the protein folds, how it changes the motion of individual parts of the polymer. And so it was really, really awesome. And I think my original job was to make the protein that goes into the spectrometer, but pretty soon I got pulled into the analysis side of things. And so Dr. Logan was, really amazing early mentor. And right next door to that lab at Florida State was the National High Magnetic Field Lab. And there I started working with Alan Marshall, who's one of the co-inventors of Fourier transform ion cyclotron resonance mass spectroscopy. So it's a mouthful, but at the time it was one of the best ways to break up polymers and see what they're made of with mass spec, because it had high resolution and a lot of other properties that made it a nice instrument. So really it was sort of the instrumentation part of things that pulled me into the coding and some of the math behind that, pulling me from molecular biology more to the quantum and stat mech side of things to figure out what I was doing. Yeah.
So that was a handful, but I get the drift.
FTICR was the way we would never actually say the acronym, right? This was an acronym rich environment, national lab with all kinds of crazy instruments. So yeah.
And how did you end up at NYU? How did you find your way to the university?
So NYU and Quran were just amazing places. Actually, the way I remember, ending up in grad school is that Dave Baker gave me a call and said, "We want you to interview." And the way I remember coming to NYU is the same way. Leslie Greengard gave me a call and said, "Can you interview?" And I came to interview for a joint position originally in the math department and the bio department. And after I gave my talk, there was some discussion, and they moved me over to the CS department. That's where my offer came from. So it was sort of a strange joint position that I applied for, but it's actually, it's how we met, and how I got involved in CDS, and it turned out to be the right decision, I guess.
Yeah, I mean, it's interesting. NYU is a strange place in an interesting way. It sort of goes in multiple directions and tends to be sort of relatively multidisciplinary, which, as you say, is how we met at the Center for Data Science. Cool. That's really interesting. So, Rich, before we get into the specifics of your work and what you're doing now, I can't but help ask, when I think of COVID and just like the phenomenon that's sort of raging all around us, infections go up, they go down. Are we like in this new normal where we're just going to have these new kind of pathogens coming out of nowhere that we need to develop drugs for really quickly? Like, is this the brave new world we're facing, or do you think this is just one of those one of a kind kinds of things, and things will settle back into the old normal?
Well, I think the way that we live, our cosmopolitan, reasonably high standard of living lifestyle, I mean, we're just surrounded by miracles every day, right? I mean, how on earth on this, like the 10th floor of an apartment building in Manhattan, do I just turn the water on? And all this power flowing into the building, the cable that goes into that building must be the size of your leg, So when it comes to the population densities we're at right now, many technological miracles have already happened in the last 150 years, right? We had to figure out germ theory and completely change the water table in almost every country to get rid of malaria in North America. We invented, at this point, I don't know, it's over 15. It's something like 19 or 20 or 25 classes of antibiotics. Each one of these antibiotics is this huge effort, right, to make essentially not just a drug, but a class of drugs. So the fact that we're at such high population densities and we have these really vibrant cities right now already relies on a lot of agricultural and biomedical technology to keep us from having to deal with infectious disease and starvation on a daily basis. And COVID is a particularly tricky one, right? We've seen these these plots where they plot the danger of the pathogen, and then on the other axis they might plot how infectious it is, or how communicable the disease is. What is its R naught? And there have been some strains of COVID that are fighting it out with measles, right? And so this is it's a particularly tricky pathogen, but I think it's important to remember that there are all kinds of things that have reduced infant mortality and reduced the degree to which we have to interact with infectious disease in our modern life, including a whole array of antibiotics and vaccines that currently exist. So I think when it comes to COVID, I think we will find a way through biotechnology and through medicine to fix it or to mitigate its effects on our society. I think for me, the troubling aspect of this right now is just that there is a growing public distrust of science that might inhibit our ability to fix this problem, right? And so that's an issue that I can't speak to as well as others. But that might be actually the key, and not as much as the technology behind the vaccines, right?
Talking about this growing public distrust and COVID, because there was and probably still is a big distrust in, like, do these vaccines really work and all of that. But if we go back to when this all started, I'd never heard of Moderna, like, as a company, and they came out of nowhere. One would expect that these vaccines would be developed by traditional pharma, and some of them were, like Pfizer. But a company like Moderna just came out of nowhere. And when I looked into it, I sort of, I use it as actually a case in class discussion. I realized that there was an old pharma and there seems to be a new pharma, right? There was an old way of doing things, and now there seems to be a new way emerging. Like with Moderna, the first drug they came up with went into clinical trials, in under 3 weeks, compared to like SARS-2, where it took 20 months, right, for the first drug to go into clinical trials.
Yeah.
So what enabled this? So tell us a little bit more about sort of this new way, the old way versus the new way of drug discovery.
Well, I think with Moderna, there's two kind of things to think about, right? How are we going? How can we, as a field and as a society, make drug discovery substantially cheaper and more reliable. And those sorts of technical questions could revolve around integration with machine learning, high throughput biology, understanding what part of human biology you want to modulate. There's a lot of technology happening right now that I think is going to make the process of drug discovery, well, substantially change from before and substantially more efficient and more effective. We're going to get better medicines, but we're also going to get them with less investment. But when it comes to Moderna, I think there's also this really beautiful story of basic science and the dream of some scientists in hospitals and public institutions developing an idea and then getting some support from investors in a startup or a venture mode, and then finally partnering with a larger pharma company to really take that drug or that medicine through the final, in this case the vaccine, through the final stages of developing all the data and the practice for dosing and for safety and all these other things that large pharma have a tremendous amount of experience with and a tremendous amount of wisdom, actually. So this was actually a really wonderful case where it worked the way it's supposed to. The investors in that sort of middle high risk venture stage, they take big risks, but they also get paid off. And then the balance is that they've done something new. They've moved the field that would otherwise be bigger and slower and have a lot more long-term strategies that they're developing that might slow them down when something new comes along. And so I've seen a bunch of cases where, not just Moderna, where a drug target that was a bit risky turned out to be a good thing to try to target. A new way of making vaccines, it's amazing, right?
Yeah, I sometimes wonder, or the question I ask is, did they just get lucky, But then the other side of it is, that luck favors the prepared, and and they seem to be sort of prepared for something like this, in the sense that they seem to have worked on developing the technological infrastructure to make drug discovery faster. I believe they described it as a tech company doing biology, as opposed to a pharma company, doing technology, which was an interesting way of sort of positioning themselves.
Yeah, and the Moderna and how we approach some of these new ways of making vaccines. This is just a beautiful story. In my own experience is actually similar in the sense that we've had a lot of ideas over the years that were really just a little too weird or a little too wild. And some of those craziest ideas we had to try out in a purely basic science environment, like a university, and others we were able to develop in a startup culture or a startup mode, like Prescient, for example, the company that led me to joining Genentech. So this is something that I've experienced firsthand too, and it's nice in the sense that you get some time to prove that you're right. You get to be wild and try something really new that has a fairly large risk profile. But you have the intuition that you're right, which is something that happens in science very often. You can't prove it yet, but you know the experiments you need to do to prove it. So it's actually a fairly well posed problem, but it's still pretty wild and there's still some risk. Well, if you're a large company with a lot of people that rely on you to manufacture medicines, and a lot of financial institutions that are tied up in your workings, you've got all these different connections. Well, it makes sense that you don't want to try something that's too wild and woolly. But if you have a whole ecosystem of startups that are, or other, subdivisions of the company, perhaps, or accelerators, well, then you can try the wild and woolly ideas and actually see what survives the Cambrian explosion. And I think that's actually, a really important part of how we invent new things in biotech today.
So let's talk a little bit more about that, right? So let's go into that, because you mentioned Prescient, which is your company that was acquired by Genentech. So say more about what led to Prescient and all of those wild and crazy ideas that you were trying in the lab that looked like, hey, these might be ready for prime time, and and your sort of journey so far into Genentech and what this whole ecosystem looks like of drug companies, and especially companies like Genentech, which actually started as genetic companies as opposed to traditional pharma, and this integration with sort of academia. By the way, this is a space I've always been fascinated with. In fact, many years ago, two of my earliest investments were in Cetus and Genentech. So that was
Really?
So I go that far back. I was like really, really interested in Cetus and Chiron and Genentech. So it's a space I've been fascinated by for a long time. But tell us more about prescient, wild and crazy ideas. How you connected to Genentech and what's going on in that space right now.
Yeah, so some of this, Vasant's actually a bit of a shared story in the sense that while we were building, the Center for Data Science, version 0.0 at NYU, we were bringing together a lot of really amazing talent. And some of that talent included Kyung-Young Cho, who is just an absolutely fantastic natural language processing, machine learning innovator who's invented a lot of things over the years. But really, when he came to us at NYU, was not very strongly interested in biology. Well, over the years, we got to know each other a little bit better, and I would say about 6 years ago, we started to talk with one of the postdocs in my lab, one of who very soon became one of the senior scientists in my lab, Vladimir Grigorovich. Well, Vlad wanted to use machine learning to integrate different streams of data to predict how proteins function. Very quickly, we got to the point where I said, if you want to understand this with any better precision or resolution, you're going to have to start thinking about what these molecules actually look like. You're going to have to start thinking about the structure of these molecules. And that, of course, takes you from the machine learning of characters and descriptions, functions, and ontologies, and all these well-controlled data structures into the messy space of atoms bumping into each other in this wild thing called water. Water is a very complex thing, right? And so if proteins are flying around in water, getting bombarded, how does any of this work? It's a nightmare, right? So, but with Kyung-Young and with Vladimir, we decided to try to put these two worlds together. The world of generating sequences the same way that we do when we translate texts from one language to another. How do we merge that with our thinking about the actual insanity of three-dimensional molecular structures? Big ones, too. Proteins are big molecules. So we tried that, and I would say, it's hard to remember the precision, with any precision, the timeline of when things worked versus when they were published. But somewhere on the order of 3.5 years ago, 4 years ago, we started to get some results from these methods that were just too good to be true. And so my role became crazy reviewer number 3. I would just say, this can't work. Have you tried that? Why aren't you citing Johnny? Why aren't you citing Sally? Like, this is ridiculous. And they would come back very soon after with actual proof that I was wrong and that it was actually working. And we went through many rounds of this, and then we realized that we were really onto something. This merger of natural language processing and structure had a lot of potential. And so we pretty quickly formed a company and actually filed for some protection on some patents, provisional patents on some of the key items that we had invented. And at that point, we realized we were in a very good position to commercialize some of this. And so that was Prescient version 0, the 3 of us. We very likely waited too long to start the company. So we had quite a number of prototypes that were in different ways being protected and published and released and illustrated with demos and things. But other than starting the company a bit too late, we had something pretty interesting. As we went out to the world and tried to find our first clients, our first pharma partners to work with, we actually found that a lot of the people that we talked to then asked us if we had a job. And that's how we got Prescient version 2. We talked to, we'll just call them Pharma A and Pharma B and that sort of thing for now, but we talked to Pharma A. And we got Stephen Ra, who is now our head of Frontiers Research, along with Kyung-Young. Absolute genius. But he was a potential client, who then asked us for a job. Andy Watkins, Simon Kilo, and others followed. And so we realized that there was a lot of excitement on the ground level. A lot of the people that we talked to also started to explore acquisition and investment and things beyond just interacting with the technology, but actually wanted to interact with the team. And so that was Prescient version 2. And then Prescient version 3 was we had a conversation with Aviv Regev and others at Genentech who are really trying to, they're not trying to, they're changing how Genentech does drug discovery. They're bringing in more data science, they're bringing in more high throughput biology, and they're also continuing to develop the world-class molecular engineering that's been going on at Genentech for 20 years. So we talked to Aviv and we talked to the business development team at Roche and Genentech, and they were a cut above. And it wasn't very long until we got to Prescient version 3, which is we're a department at Genentech. And that was a scary transition. It was a big change of plans for us. But I do think we made the right decision, and that we can actually develop this idea a lot faster by integrating into this, top class, top tier company, Genentech. So this has been amazing.
So wow, there's a lot of stuff in there. As you were talking, I couldn't help but asking myself, what's the linkage between language models and biology, right? Everyone's talking about ChatGPT-3 now, right? I mean, you can't go any place without people talking about ChatGPT-3. And I remember Kyung-Hyun's recruitment talk. I was there, and he was talking about all the work he'd done about translation from French to English, because documents in Canada all in French and English. So, what's this connection between the language work and biology? I mean, is that something you can reduce to sort of a few sound bites?
Yeah, I think so. There's two really amazing things that we can appreciate when we think about biology. Like one, and again, to some degree, a picture would say 1,000 words here. So I apologize to the listeners, but imagine just a tree that's bigger than any tree you've ever seen, a tree that stretches to the moon and has a lot of branching. Well, this is the tree of life, right? You have all these organisms, and they're reproducing or not. Some of them are going extinct. So a lot of the branches we'll never see. But within those branches for the organisms, there are also other little sub-branches within that are the branches for the genes. The genes themselves are evolving. And so we have versions of molecular machines that have existed for a very long time, and they persist to the current day. And they have different versions, different battered up, beat up versions of these molecular machines in lots and lots of different organisms. So evolution and comparing molecules that do similar things, genes that do similar things to each other, is always in biology. From the beginning of computational biology, it's always been a treasure trove of information, and that information can be turned into praxis pretty effectively. Well, this is a natural hunting ground for Vlad and Kyung-Hyun, right? Machine learning that powers a lot of the natural language processing, speech and text and image stuff that we do in big tech. A lot of that can dig into this gigantic treasure trove that is all the sequences we know about. And then with structure, now again, we can approach structure in a lot of ways, but we can use machine learning as a way to deal with one key problem, which is that if you have hundreds of parts of your gene, you have a long string, a polymer, it has to fold up to form a compact three-dimensional structure. You just have a lot of different bond angles that can rotate. Imagine the protein as the world's biggest Tinker Toy assembly that you've ever seen, this gigantic twisty toy. Well, twisting all those bond angles turns into a nightmare, a real nightmare. You might not wake up from this one. This one is just like, eons of twisting those bonds before you get the right answer if you do it wrong. Well, machine learning is an absolute miracle when it comes to that optimization problem. How do you find the right answer? How do you look at other right answers and map some of it that generalizes, but not too much so that you're memorizing and getting the wrong answer? Machine learning helps us navigate that space of structure really, really effectively. Part of it is how we score things and decide what we like and don't like, but part of it's also just how we navigate that really complex space of possibilities. Okay, and so now, the last reason that it's important to have these new technologies in the mix is now if we imagine we have the world's biggest tree and the world's biggest tinker toy, geometric toy assemblage, and we've used machine learning to figure out some ways of dealing with these two things. Well, how do we put our wisdom, our knowledge, our experience from these two very different ways of looking at the world together? And so the last kind of, maybe the third way that we could sort of describe without too many pictures is just that, when you have very different ways of looking at things, one is the sequences, the parts, the bases, the amino acids, the characters that make up evolution, the things that get inherited from generation to generation, well, they also are alive as three-dimensional structures that wiggle in solution and do all kinds of wild geometrical things. But both of those realities exist. Right? Evolution is happening at the informational level. And those things that are evolving at that character level, they really are three-dimensional things that are wiggling around. Both of these things are happening at the same time. So to reconcile those two realities, machine learning gives us a great number of machineries and methods and even some theories here and there. And so that's that integration of those two ways of looking at the world, which are both definitely happening at the same time. That's a really important part of what we're doing at Prescient. And of course, we're not the only ones, right? There's hundreds of groups doing interesting things where they're taking advantage of that integration, evolutionary and sequence, with structure and molecular function. It's really, really wonderful time to read the literature.
Yeah, and thanks for that wonderful imagery, Rich. I know a picture is worth 1,000 words, and I've seen some of your pictures, and the pictures themselves aren't straightforward to understand, but, you sort of put it well in terms of this sort of molecular machinery. I'm wondering, are we basically machines deep down that that are built of this sort of 4-character alphabet as opposed to a binary? Is that a reasonable way of thinking about what we are, or am I reducing us to something way too mechanical? People talk about free will and all of that stuff, but does it ultimately come down to machinery? Programming?
Well, we definitely have a lot of pretty amazing molecular machines in each cell. And then how the cell decides what molecular machines get to play in any given cell, there's a whole informational or regulatory level there, kind of surrounding the genome. What parts of the genome get made, what don't get made. And then when you get to the brain, well, those cells actually get tangled up in a lot of really interesting ways into actual cell circuits. So that, the central nervous system is definitely a very interesting, complex system with a lot of different scales interacting. The molecular, the genomic, the cellular, and many others, right? So it is quite a beautiful system, right? The sheer connectivity. Neurons also get to not just be connected in their circuit, but they actually do a lot of thinking about what they're going to do every morning. They're turning genes on and off, and they're definitely there's definitely a component of learning that's related to the decisions that are being made within the cells as well. So there's a lot going on there. That's not as much my specialty, but it is definitely something that I like to think about, and I like to think about what happens in those neurons kind of at the molecular level.
Thanks for that summary, Rich. So, you've been talking about machine learning, where we think about building models from data. And traditionally, we've used carefully curated data to build models. But more recently, models such as ChatGPT and Envision use all the text and images out there to learn in a less supervised manner, which some people call unsupervised or self-supervised. Tell us about which methods from machine learning apply to biology and what the data look like for training.
Yeah, that's a great question. Well, the first thing to describe is just where we're at without the machine learning. What is happening in the gigantic laboratories that are associated with universities and large pharma, and there we're just living through an amazing time with respect to high throughput biology. In 20 years ago, we maybe had 80 genomes, and now I don't know how many we have, but if you include bacterial genomes, which one should, we have millions of genomes that are fully sequenced and lots of pieces of genomes as well. So there's been a huge explosion in what we could see in the world of sequence. And so there is a huge portion of what we do in biotech where unsupervised approaches to the full corpus of data is the first step. And so there are many things we do where we look at databases that contain billions of sequences and build language models and other types of models that can think about sequence and structure in a self-supervised way. That is definitely one of the key drivers. We do quite a bit with supervised learning as well, and there are actually a smaller number, but still a pretty substantial, pretty amazing number of protein sequences that have their functions carefully annotated. And some of those systems have been being diligently developed for decades. Some of those systems, like CAF, they're databases that control hierarchies of how we describe protein structure, the shapes, the subpatterns, the so-called secondary structures, which are sort of smaller motifs within those structures. We also have databases that talk about functions that we might understand a little bit more if we have a chemistry background. These are things like enzymes. They take in a molecule and they modify it. They take in two molecules and they put them together. They take in one molecule and they break it apart into two other things. Enzymes are important for our energy, the synthesis of the parts of our cells and our bodies. There are other, and then the last kind of set of annotations and supervised learning that we might have would be the huge amount of information about the information processing part of the cell. And again, a lot of the information processing that happens in cells happens via proteins that facilitate that information processing. These are proteins that turn on and off parts of the genome. These are signaling proteins that take different stimuli from the outside world and use that to modify chemicals within the cell. So there's all kinds of things going on in the so-called regulatory and signaling pathways within the cell. Those things are organized into networks. They're not as much organized into labels and ontologies of function, but just very simple connections between very large groups of protein families. But there is in biotechnology a huge amount of supervised learning that goes on, with those being three of many different types of annotations that we use. At Prescient, what we do is we always come up with we typically come up with generators that say, how can I change the protein? How can I change the molecule that I'm designing? And so the mutability is something that is in some ways completely flexible. You can string together any set of amino acids you want to. But the goal here is to make as many changes as we can without breaking the fundamental essence of a particular protein. So we might be interested in an antibody. It's a part of your immune system. It's how your immune system recognizes pathogens in some cases. But it's also something that we see across all mammals, and we can take those domains, those antibody domains, and we can re-engineer them in different ways to make medicines, to make molecules that are actually doing something that your immune system didn't do, but still has a benefit to your health. It attacks an oncogen and facilitates the killing of tumor cells, for example. Well, as we're engineering those antibodies, we don't want to break them. We don't want to unfold them. We don't want to make them get cleared from the bloodstream too fast. We don't want to diminish these really wonderful properties that evolution has conferred to the protein family. And so we can change the things that might change how the antibody binds other things, but we don't want to change the things in the molecule that cause the antibody to fold. So we need an elaborate system that tells us what changes we can make and what changes we can't make. And we need that system to be as fast as rolling the dice in the computer. So that's what the self-supervised part does. It's a system that says you can make these 13 changes. You can make these 5 changes. You can make these 2 changes. And every time you roll the dice, you have to tell it 2 things. How wild do you want to get? How many changes? And that's kind of it. You tell it how much to wiggle. And sometimes you can tell it which direction to go in. This is something a little more complex we can get back to later. But you give it a small number of parameters, and then it gives you a cassette of changes. And then it's up to you to decide with a different layer in the machine learning whether you like those changes. But that's what self-supervised learning can provide. It can say, I have seen every antibody that exists, millions of different antibodies from all kinds of different databases, contexts. And so I can tell you changes that will keep this protein and antibody. Now, whether those changes make it a better binder or a better medicine or drug, these are things you have to figure out with other pieces. But I can tell you how to be an antibody, and I can use that as a map that allows you to mutate the sequence substantially to come up with new functions to layer onto that molecule. But you can do that with the insurance that you're not breaking the molecule, or that you're breaking the molecule very infrequently. Now, if we were to take random mutations and just start wiggling the sequence, substituting one amino acid for another, one base for another, we would most of the time break the sequence. Most of the things that we do in evolution have to be compensated by other mutations very quickly, or else we break things. And the goal is with machine learning to get to a point where we can actually only make mutations that are safe.
Wow, a lot of stuff in there, Rich. So, you talked about sort of the unsupervised and the supervised, the unsupervised being the sort of self-supervised learning, which has been incredibly successful in language models, right? Where you say, you get the machine to predict a particular word in the sentence that you masked, right? So,
yeah, that's right.
And it learns to predict that really well, just looking at the entire corpus of like written stuff out there. So if you say the cat blah over the fox, well, it's probably jumped or whatever, and the machine learns to predict each word in a sentence. So that's sort of self-supervised learning. Where exactly is the equivalent here? Like, are you trying to say what kind of mutation can I make that, and what's the prediction problem? Is the prediction problem to say, what's the missing change I can make to a protein? And is the prediction, will it still fold in a certain way and have certain desirable properties? Like what's the connection between sort of the self-supervised learning in language and the kind of self-supervised learning you're doing in biology?
It's very similar to what you just described. Here, though, instead of if we have the cat jumped over the brown fox and we hide jumped, well, here we would have a sequence for an antibody, and we would hide a handful of amino acids in the middle of that sequence. And now those amino acids might be pointed to the middle of the molecule. And so we want to substitute other of the hydrophobic or the greasy amino acids there, and we don't want to substitute a really large, highly charged group that would blow up the protein. So it learns to not stick an arginine there, which would be one of those residues that's highly charged and might blow up the protein. And instead, it learns, well, there was a tryptophan there, which is a big greasy amino acid. I don't know if it has to be a tryptophan, but I would think that these other 4 amino acids would be the right ones to put there. It learns that from the positional cues of the surrounding sequence. And once you learn that, then now, of course, when you're training, you're hiding or masking, or you're doing different things, different tricks to teach the molecule how to fill in the gaps. But then when you're designing, you could say, well, I know that these are the residues that are touching the target protein. The target protein is a human protein, and we want to design a medicine that binds it very tightly. Well, I have a model of how the antibody is approaching that target protein. From that model, I can figure out which are the parts of the molecule, the antibody molecule, that I want to change to improve that affinity, to improve that specific binding to the human target protein. But while I'm changing those amino acids, I don't want to do anything that might have a collateral effect, an accidental effect of breaking the scaffold that loop or that region is on. Because remember, the antibody is still a very big molecule, and most of that molecule is busy keeping the molecule together. Only a small subset, something like 10 or 20% of the molecule, is involved in binding the target protein. And it's not always trivial to know who's involved in binding versus who's involved in folding, because if you fold better, you might bind a little better. And there might be a position far away from the binding site that has an indirect effect that still improves binding. And so it becomes a little, well, it's non-trivial to figure out how to separate out binding from folding, for example. But you need both. And so what we want to do is we want to mask the regions and fill them in And that process then isn't just denoising the sequence, the biosequence, but it's showing us all the alternates that we might try, all the alternate sequence modifications that we might try. And then we can do that process over and over again to generate lots and lots of alternate sequences, which can be fed into downstream processes. And now, of course, I'm describing this not just in a very general, why is unsupervised learning part of this process? I'm not necessarily describing exactly what we do at Prescient, because you can imagine that you set things up with the self-supervised is generating mutations, and then you have other supervised methods that are deciding which ones work. But people innovate and they find ways to integrate those processes. People innovate and they come up with new ways of, instead of using denoising autoencoders, using diffusion processes and other things to compose the core models of these methods. And so there's a lot of innovation such that we've drifted a bit from that really clean separation. But there is a self-supervised component to nearly everything that we see in biotechnology for molecular design these days. Yeah.
Fascinating. Sounds like the machinery is focusing the search for hypotheses based on the available data at the genomic and molecular structure level. Is that right?
That's right. That's right. And the hypothesis would be that, this mutation isn't going to break anything. Yeah.
And who's the teacher here? And so when you try this thing out and you say, well, here's what I predict will be the property of this mutated molecule, where do you get the supervisor part of it from? Because in language, it's sort of automatic. It's there in language and it's in the sentences. What's the equivalent here in biology? Who's the teacher?
Well, so biology is a field where we've spent a lot of time over the last 20 years organizing databases, ontologies, and other systems for keeping our data, not just fair, but also really nicely accessible and hierarchical, through hierarchical methods and things like that. So biology's done a lot of work here, and depending on the problem, there's a lot of publicly available data. At Prescient, we're using a lot of high throughput data that comes from different stages of the drug discovery process. A key data point is whether or not a potential medicine, a potential molecule that could be a potential medicine, binds the target specificity. And we call this affinity data. Now, if you're thinking about T cell receptors and cell-based therapies, you might instead of thinking about the binding strength, you might think about what happens to the cell downstream. You might think of activity. If it's an antibody, you might be mostly interested in the specificity of binding, so-called affinity. And if it's a small molecule, you might be, depending on the assay, you might be thinking about binding strength, but you also might be thinking of potency, which is a combination of binding strength and activity. And it is something that differs depending on the assay that we're using. But whether it's a cell, an antibody, or a small molecule, that key parameter we have an excess of that kind of data in any large pharma company. And so that's the data that can be used to supervise these models. There's some of that data in the public sphere, but mostly you find that data in startup companies that specialize in those high throughput assays, or in large companies that have multiple decades of that kind of data accumulated in their informatics systems. So it's been really wonderful developing these models at Genentech, where we can rely on the informatics teams who can really help us with the care and feeding of some of these really complex models. Yeah.
So from the perspective of university industry partnerships, using Prescient and Genentech as your case study, where's the synergy in such a partnership?
Yeah, so, for us, there's lots of ways in which we get a lot from our interaction with Genentech. One of the ways is, like I just mentioned, the data that we need to train models for drug discovery has to come from a real drug discovery process. You can try to train models outside of the realm of a proper drug discovery company. So say more about
that. When you say it has to come from the drug discovery process, say more about that. So what does that actually look like?
Well, so I can say some about this. I can't dig too deeply into the actual way in which we do this. But one thing to point out is that we have to think about the structure and the biophysics to some degree, right? We have to make molecules that bind specifically to the intended targets. And for that problem, we can look at a lot of public data sets. But if we use the data accumulated in Genentech and the Roche family, we have substantially more data. And so that's a case of, quadrupling the size of the data that would be accessible in the public sphere. That's very important for our machine learning methods. So that's one way. The other thing to think about is that there are lots of properties that molecules have to be medicines that are safe and effective and that help patients. And some of those properties are things that are really not as widely available or even at all available in the public domain. Those things include how long the drug stays in the bloodstream or in the site of administration, so to speak. They might include the way in which mammalian or primate immune systems interact with the drug. If it's going to be an effective medicine, it can't be something that you develop an allergic reaction to, which, again, this is a bit of an oversimplification, but and then there are a great number of other properties as well related to formulation and packaging of the drug and all these sorts of things. But you have to get all that right at the same time. So what we want to do is make machine learning methods that when they're doing that early part of just targeting the human protein of interest, you have all of those other babysitters active so that you're not making this beautiful binder that's going to really piss off the immune system. You're not making a beautiful binder that's going to crash out a solution or do something else that's quite terrible later on in the formulation or packaging stage. So, in order to do this right and really take advantage of what machine learning can offer, you have to have access to all of those different data types, and you have to have access to those data types derived from the assays that are part of the practices of that particular organization. You really do need to integrate deeply what you do. So we have that advantage. But then, I think the most important thing, right? Those are technical advantages. Those are data getting to our models so that we can make models that are actually useful to Genentech. And that has to be a give and take, and that has to be iterative. But there's a whole nother level that sort of supersedes this, which is we have to, be at a place that innovates not just in the design and the engineering of antibodies like Genentech, but we also have to be at a place where the selection of targets, the biology and the thinking about what you would want to change in a human system or in a system to affect a positive change for a patient. This is a really complex process. And so for us, knowing that when we're focused on our molecular design module, that we have colleagues who actually know what to change in the first place and how this fits into this holistic picture of the human organism and the patient. I mean, this is a very big can of worms. There's machine learning and statistics and data science involved in that, but this is where the biologists who are going to drive the vehicles we make around, this is where they live. And so we want to get to know what their specifications are. We want to get to know what are the demands from that side of the business, and we want to make sure that we're making things that are responsive to those needs as well. Because if we don't, then we're making something that's a very powerful module, but it might have an impedance mismatch to the rest of the system. And it might be, it just might be solving a problem that's not the rate limiting step, right? And so for me, that doesn't help me make better molecules, but it helps me motivate the team and justify what we're doing, because we know that at a place like Genentech, we have the best-in-class selection of what perturbations we should be thinking about as molecular designers in the first place.
So as you were talking, I realized how messy biology is. Actual outcomes of interventions need to be carefully measured, and the process of drug discovery is guided by this kind of data, which, in the absence of simulators, must come from the field.
That's right. Yeah, no, you summarized that pretty well. And so, yeah, exactly. It's as scary a problem as molecular affinity, right? I could scare you with there's billions of years of evolution and billions of sequences and all these degrees of freedom make it an impossible problem, but somehow we're able to engineer affinity anyways, right? Well, you can imagine we've broken the problem down and worked on it for 20 years. And there's ways in which we break the problem down and then approach it with machine learning, and we're able to build these molecules from the ground up, and it works. Well, on the human biology side, there's just an amazing concentration of effort and talent that goes into that as well. And the key, like all these things, is to break it up. So one part of it is human genetics. We have genomes, sequencing and exploration of genetic variation across lots and lots of folks. And so we can actually look at different diseases and different parameters in the sort of overall health ecosystem and see what are the relationships with different changes in the genome and different genotypes, so to speak. And sometimes, not always, but sometimes that can suggest a target, that can suggest something that we can modulate to help a subset of patients. You can see there's too much of this gene, or too much of this gene hurts, too much of this gene is protective. Missing this gene means you never get this disease. So maybe knocking it out would help patients at a certain point. So there's human genetics. There's also a lot of what we have to deal with as we try to improve health that is conserved across the tree of life. And so we can actually look at other organisms, and we can look at model systems to learn a lot about things. And then also there's lucky for us, there's sort of a different set of things that we need to do across different diseases. And so in oncology, for example, some cancers, the cells that are dividing too fast they've also carried along some accidental changes that make them different from other cells in ways in which that allow us to target them or identify them or interrupt their function. And so there's a sort of a spectrum of difficulty. Sometimes diseases and indications that we want to go after have evolutionary accidents that we can exploit. And so the teams that work on what to do, identifying targets, they have all kinds of ways of breaking this problem up with animal models, human biology, human genetics, and even genomics and molecular biology. High throughput biology plays into this quite a bit as well. It's really something. It's a big effort in academia and in pharma, and it's really another place like molecular design where it's just a great time to read the literature. You're seeing so much happening at so many different places. That it's sort of driving this new holistic understanding of human biology. It's really, really quite amazing.
Fascinating stuff. So I guess traditionally pharma companies used to be focused on, let's say, solving a disease, right? That was their target. Now it seems to me that the search has become more general than that. It's not just about targeting a disease, but actually building a capability that's, where you have the ability to create drugs rapidly, in response to, any kind of target. So is it a change in way of thinking at these pharma companies about what drug discovery means? Is that kind of what machine learning is bringing to the table here?
Well, if we focus on the module that Prescience focused on, which is making it very reliable to design high potency molecules, there are a lot of ways in which that can change how drug designers work, how antibody engineers work. And I could give you just an overly technical example, but I can give you one example that illustrates a little bit about how I think about things. Well, the first thing to do, again, we have to paint a little bit of a picture. So what are we doing when we're designing medicine based on an antibody? So let's imagine we have a receptor on the surface of the cell, right? And so the way I want you to imagine that receptor is it's a beach ball with a 2-liter bottle hanging off the bottom of it, a 2-liter Coke bottle. The Coke bottle is pushed down into the membrane, and it attaches that beach ball to the surface of the cell. For now, that's all we have to imagine about our receptor, just its shape. Well, traditionally what you would do is you would take just the beach ball part of that. You'd leave the Coke bottle behind, and you'd express just a fragment of that protein at high levels, and then you would inject that protein, just the beach ball part of it though, into a rodent. And the rodent would just kind of go about its business for 2 weeks while you occasionally infuse this protein into it. It might, its immune system is going to fight off the beach ball. Later on, you can then pull those white blood cells back out of that rodent, and you can figure out which ones seem to be really quite common compared to the rest of the background, and you can find things that bind the beach ball. Right? Now, that's just the beginning of a whole process, right? Because remember, I said before, we still have to make these things into medicine, so they still have to go into humans, and they still have to not really irritate the immune system, and they have to stay in the blood of a human, not a mouse, for a long time, right? So that's just the beginning of a long road. But having things that bind that beach ball part of the receptor, this is the beginning of making a medicine that targets that receptor, which might help this disease, beach ballitis or whatever. Okay, but that if we go back to the actual biology, that receptor might interact with other receptors. It is doing things in that cell. And so if we imagine that how that antibody binds, it's going to bind to a fraction, 20% of the surface area of that ball. And depending on where the antibody binds, and there could be all kinds of ways, different ways, you could bind on the left side, the upper side, the bottom side, close to the membrane, et cetera, et cetera. Depending on where on that ball you bind, you might have different effects on the actual biology of the cell. And that's important. And so if we can paint part of that surface and de novo, from scratch, design an antibody, well, this doesn't mean that there's less work for the folks in the lab. It just means that now, instead of worrying about binding, they can focus on activity. And the activity is still a very complex piece of biology. It's still going to require robots that handle cells and growing things and doing wonderful molecular biology, having to work through pretty complex logic to figure out what your data means. All the good stuff that is molecular biology, it's going to get, well, it'll be about as complex as it ever was. But now, instead of having to take random things from this repertoire, we might be able to purposely design things with different geometries so that we can build a set of molecules that allow us to better explore the downstream activity and the real effects on the cell. And so I think we're hoping that we can make it easier and more reliable to design molecules so that we can drive the combinations of different functions, the exploration of activity, and the exploration of all the downstream cellular biology. Now, at the same time, our colleagues are also applying a lot of really high-tech stuff to that molecular biology. So hopefully we can build a bridge somewhere over the void there and come up with things that have more complex and more multifaceted activities and effects on cells. But for us, that's how we think about why we're trying to make it automatic to design affinity, because that's going to allow our colleagues to really explore activity and the effects of geometry and the effects on these things that we're designing with greater depth, because then they can skip step one and get straight to step two, for example.
Great stuff, Rich. When you were talking about things like geometry, it makes me realize that in biology, it requires a solid causal understanding of what's binding to what and why. And there are actual mechanisms involved that the machine learning part must somehow deal with. There's sort of a structural aspect involved that's more complicated than traditional machine learning, isn't it?
Yeah, that's right. When we do machine learning in this larger molecular biology context, right? And whether it's for basic science or drug discovery or for ecology, right? We have to make sure that the machine learning powers the discovery of mechanism, right? Because machine learning, in some cases, it encapsulates mechanism, and then we don't have to learn it, right? There's, this famous, Socrates and Phaedrus had a lot of discussions, but, one of them was that, the power of the written word, right? And, there's the famous argument that by writing things down, you're not actually creating a memory, you're creating a reminder, and then the person who has that reminder has less memory, right? So does machine learning, like the written word, essentially replace or subvert an important human mental activity, right? And I think that when we do it in biology, like I said, whatever we're doing at any scale in biology, those other scales above and below are still there. And so we have to think about machine learning empowering the discovery of mechanism so that we can then stitch those connections back with other layers of scale that we have to deal with. And so here, one of the things that we think about is that the machine learning can focus on binding. And we know the mechanism relates to the physics of molecular interactions and atomic interactions in a condensed matter state, in a very dense state. We know it revolves around the physics of liquid water, which is quite complex. And so all those mechanisms are understood in principle, but then bridging the complexity of all those interactions compounded in a very large molecule in a hydrated state, that's a bridge too far sometimes. And so we sort of know what we're doing there. We're encapsulating known mechanisms, but we're dealing with a scale issue in some regards. Now, there are other things that we're also doing. We're probably making a lot of corrections to misparameterizations. We're also approximating a lot of things that are true n-body problems as triangles and pairs and subunits and things like that. And so there's some approximation, and there's some encapsulation of mechanism, and there's some addressing of scale. But when we do that, we hope that when we hand things off to the next level, we've also decoded a three-dimensional structural model of what's happening. And so a lot of how we deal with this question at prescient is to try to make sure that we understand our inputs and the mechanism applied to them, even if we're encapsulating in some ways that are difficult to peer into. The other thing that we try to do is make sure that we hand off models that have this structural interpretation, which we hope provide some ability to then connect back to activity and things. But I think it is a really good question. The other thing that we have to think about when we think about mechanism and making sure that our machine learning empowers as much mechanistic understanding as we can, as much encapsulation across scale as we can. I think this also brings up a lot of questions about decision making and active learning and experimental design and error estimation. And that's a big focus at Genentech right now as well. And so here the question is, we know that we're going to use the machine learning to queue up experiments which can then triangulate these mechanistic conclusions. Well, how do you do that? Right? A machine learning method can probably sample 1 million sequences. It can probably, the inference times and the efficiency of these methods are quite amazing. So that kind of means you can make even more designs. And so you can overwhelm what the robot can handle. You can overwhelm what the self-assembling wild DNA machine can synthesize. Even if you're using the most advanced high throughput methods, you still have to think really carefully about how to populate those methods with the best guesses from the machine learning method. Because in biology and molecular biology, we use machine learning most of the time to queue up experiments, which will then get us back to that mechanistic understanding.
So, Rich, what kinds of partnerships are we likely to see between government, industry, and academia going forward in this space?
Well, this is a big question for me and something that's been on my mind lately. Right? I am the product of national labs and universities and benevolent professors who have, taught me a phenomenal amount over the years. But currently, some of my favorite ideas I'm developing at a large company that is, providing the resources, but also providing a substantial amount of program project management skill and the sort of skill to interface these with the needed downstream methods and groups of people. So it's not just the resources, but a huge amount of organizational power as well. So I am a product of a university that's doing something currently that I could probably not do at a university. So to me, the answer to this question is a bit entwined with some existential questions right now. But I guess for me, I do see that there is an absolute need there is a very strong need for both of these sides of things, right? A lot of what we're doing at Prescient came out of a lot of casual discussions at a university that then became pretty open-ended explorations of a handful of ideas, right? We're thinking about diffusion models. We're thinking about self-supervised methods for denoising sequences. We're thinking about optimization methods that are actually pretty new. A lot of the modules and pieces of code that we're developing are being developed by people who were pretty recently trained as PhDs. And a lot of the methods that we're using actually came out of pretty open-ended basic science explorations. So I see this huge need for this. The other thing is the academic world is a world where sharing is the norm and transparency is the only way in which you can get recognition for your ideas. And so it doesn't just drive innovation, but it's also a place where a lot of dialogue can happen, including dialogue between different industry partners, right? There's a lot of ways in which industry probably indirectly shares information through these academic collaborations and through sort of cross-fertilization of people within these academic and sort of not-for-profit places. So I see that as absolutely essential to powering what we're doing now in industry. At the same time, there are some things that we're doing in academia that they're being done in a basic science setting, but they are essentially fundamentally empirical engineering challenges. And so to really see the full potential of some of these technologies, you do need to use this modern large company structure to put together a big enough team to actually function and develop the full potential of these ideas. And so for me, I do see a lot of value in having that as a place where people can land and do science and actually test out some of their ideas at scale. So I see a lot of value in both sides of things. And the startup scale, the sort of in between those two scales and those two sets of functions, like we discussed, is a bit of a way of translating some of these ideas. But how that balances, what are the right proportions? How does one justify the other? That's an active debate. That's something that I think we need to keep talking about to calibrate the relative interest here. If I answer that question in the most practical, the most boneheaded way, I could say it's still a great idea to get a PhD. And what we're discussing is whether it's still a good idea to do a postdoc, or these sorts of issues. When it breaks down to the individual level, there's a whole range of interesting issues. And so we have to figure out what balance we need for society, but then we have to go back and reconcile that with the choices that people make as individuals. And that's when it really blows up in terms of me not knowing the answer to this question. Yeah.
So what is the most interesting space to get a PhD if you're working in this? Should you get a PhD in computer science and biology and data science? Academia tends to be siloed, and actually data science is one of those spaces that is designed to be interdisciplinary, right? So if you're someone young who wants to get into this space, how should you think about it? What's that right balance between biology and computing and anything else that is useful in this space? How should you think of training yourself?
Well, I have to say that I really do like the spirit of the Center for Data Science at NYU and some of these related programs like the Data Science Center at Columbia, for example, where we try to, and again, it's not easy, but we try to bring people from lots of different application domains together, but then provide a really solid quantitative framework for the PhDs and master students. I like that idea. But again, I think this is definitely something that has to be answered individually, right? I think for some people, they're just such gifted molecular biologists that's their specialty. They're going to develop new systems, new approaches, new molecular machineries. They're going to discover new gene systems. A focus in machine learning for these folks would be a waste of their natural talent. There are other machine learners who could really thrive in a more applied setting. There are other machine learners who are just gifted at theory and trying to take some of these hard-won empirical results and try to build some theory behind them or above them. And so I think it's really an individual question. I think for me, I have been extraordinarily lucky in my career to be allowed to alternate between what my focus is so that I can develop quantitative skills, but then really get embedded in some interesting applications and back and forth. And this new phase for me at Genentech is one about, engineering a broader organizational capability to try something at a scale that I couldn't try before. And so this is kind of a new thing for me. But the computer science, chemical physics, biology combination that I had growing up, I don't know if you find that in any one program. So I would say to anyone starting out, start with a skill, have an application interlude, and then let yourself do whatever you want to do after that. Like don't be in a situation where because you developed a skill, you think that you have to develop those sunk costs. Let yourself really try pretty dramatically different things if that's what following your heart means at that point. And then the key to that working is saying that the salary or the compensation involved in any stage in my career, it can be in dollars, but it can also be in visibility, right? When you're younger, when you're earlier in your career, you need to be able to show people what you've done if you really want to develop that later stage or middle stage of your career properly. So you don't necessarily have to get the interdisciplinary experience all in one phase, I guess is what I'm saying. You can imagine, just as long as you're disciplined about taking risks and doing things that scare you, then you're allowed to sort of spread that out over different phases.
Well put. I think this whole sort of the experience here seems to be multi-layered, and what you're saying is that individuals need to find their path, right? Whether they begin in biology or computing, but somehow the new world seems to be sort of integrating these all together. And it seems to me that the people at the forefront are people who can understand the connections between these different areas, especially the biology and the computing. But how you get there seems to be kind of, follow your individual path and what appeals to you.
Yeah, that's right. And, we talk about machine learning and technology. We talk about interdisciplinary activities and how to train people. But like Vasant, I wonder what you think about how the current set of informational tools, right? Machine learning is a way that an individual or an organization can process data, but there's also a tremendous amount of information that's more available via search and archival activities and the fact that machine learning is translating and finding and organizing things for us all the time. And so are we as scholars able to do more? And so interdisciplinary things that would have been silly or would overwhelm an individual a generation ago, maybe we can have more tolerance for that combination because it's facilitated by quite a bit of technology and just sort of institutional support for these things that wasn't there maybe a generation ago. I wonder about that. And it'd be interesting to discuss that a little bit too. But
yeah, no, that's a whole new that's a whole other discussion. I think you're right. I mean, I think the tools out there are just facilitating, more and more sort of the interdisciplinary interaction. I mean, ChatGPT-3 seems to be able to pass, all kinds of college exams. I don't know whether it'll pass a biology exam, probably, right? And I can imagine future tools where you ask a question and it'll say, hold on a second, let me just have issue this query to this database, and it comes back and it's assembled answers for you on the fly. So, it's an interesting world we're heading into. Rich, thanks for that. Thanks for the time. I really enjoyed the conversation. There was a lot in there to unpack, and, we'll leave our listeners with a lot to digest there. But thanks for your time. I think that was fascinating.
Well, thanks for the questions. Thanks for the conversation. This was great.